Hello all, and thank you for joining us on this monthly scientific webinar today. We're going to start with an introductory session on Munir Armeni's Research Center, who we are, and then we're going to give an introduction about the speakers. Munir Armeni's Research Center, for those who don't know yet MARC. MARC is a research scientific center that aims to advance science through conducting research in different disease areas. Our goal is to translate our scientific discoveries into effective medical practices for better healthcare. We believe that we share a vision and a purpose that, together with an action plan, can honor the belief through knowledge, truth, and science to help build a better humanity. In today's scientific webinar, we're going to talk about cancer research. Cancer research today is the most powerful weapon in the fight against cancer. In recent years, a new era has dawned in cancer therapeutics, offering a better understanding of tumors and the innovative strategies for a better cure. In this month's webinar, in celebration of World Cancer Month, we're going to give awareness and inspire action for a cancer-free world by giving a talk about recent research that aims to give us a cancer-free world. Today's webinar title is about cancer research, which is the powerful weapon against cancer. In session number 1, it's gonna be given and presented by Professor Ulrich Sack. Professor Ulrich Sack is the head of diagnostic laboratory at the Institute of Clinical Immunology Medical Faculty, University of Leipzig, Germany. After his medical study at Leipzig University and a fellowship of the Alexander von Humboldt Society at the clinical research groups of the Max Planck Society in Erlangen, he completed his qualification in clinical immunology, and since 2002, he has been holding a professorship at the Institute of Clinical Immunology at the University of Leipzig, Germany. From 2008 to 2015, he supported the Translational Center for Regenerative Medicine at the University of Leipzig in the function of the research director. Furthermore, he was a group leader and an advisor at the Fraunhofer Institute for Cell Therapy and Immunology from 2005 to 2015. Professor Sack is involved in several networks of scientific experts. He has been the chair of the IFCC working group, flow cytometry from 2011 to 2018, offering 16 flow cytometry courses in Europe, Asia, South America, covering basic science, immunology, and hematology. As a technical assessor, he promotes ISO 15189, 17025, and 17043 accreditations. His scientific work concentrates on cellular immunology with a strong focus on immune function and flow cytometry. He published successfully more than 300 research papers. And right now we're welcoming Professor Sack, and he's gonna start now his session. Can you share the screen now, please, Professor Sack?
Yes, I will do so. Thank you very much for the nice introduction.
Thank you so much, Professor.
Can you see my screen now?
Yes, now. You can start now, Professor.
Okay, perfect. Thank you very much for the nice invitation and for the friendly words in the beginning. And I would like to apologize for all these complicated German words. But otherwise, my Egyptian and Arab knowledge is also very poor. So sorry when I pronounce your names in the wrong way. Today, I would like to show you some results of our group in the field of immuno-oncology. First is a declaration of the conflicts of interest. I'm a technical expert, as you told. I got travel support and lecture fees from some companies, and I'm a member and the proficiency testing supervisor of Instant. Instant is one of the providers of external quality assessment schemes, also active in Egypt. Today, immunological therapy principles are changing, and they increasingly determine the modern medicine. So we have today a lot of immune therapies. Most of these therapies are based on antibodies, but also on small molecules, soluble receptors, or cells and modified cells that can be used. I show here some examples. For instance, a CAR T cell with a CAR T cell with an genetically added receptor against the CD20 antigen, or antibodies functionalized and working against tumor antigens, or inhibitors of kinases that can be used for the treatment of tumors. For these new immune-based approaches, there are many targets available, and a lot of this is still in the state of research, but some of them also are going the way into clinics. And when you see this slide, it's taken from a review work, it's cited in the bottom line. You see that certain cytokines and signaling molecules can be addressed to treat patients and are today candidates or even existing drugs. The effects of these cancer immunotherapies are hopefully an anti-tumor activity. This is what we expect. We treat the patients to treat the tumor, to defend the tumor, and finally, hopefully, to cure the patient. Sometimes these drugs stay in the patient, and this can be very relevant for the prognosis of the patient because there will be a long time effect. In addition to the desired effects, these therapies also affect and modify necessary functions of the immune system. When we modify, for instance, the generation of B cells, for instance, by applying anti-B cell antibodies, of course, we induce an immunosuppression. When we stimulate immune cells by inhibiting checkpoint molecules, we induce immune stimulation. When we suppress the immune system, we also suppress the immune response to vaccines and to infections. And sometimes we induce a tolerance failure by inhibiting necessary checkpoint inhibitors. Furthermore, all these therapies very often are combined with other therapies. For instance, initially in lung cancer patients, we start with the surgery, or we can combine immune therapy with irradiation or with chemotherapy. All these additional therapies contribute directly and indirectly, for instance, via stressors, even psychological stress, to an immunosuppression and to general side effects. Sometimes this is also a bit surprising. What is the consequence? Here you see Kaplan-Meier curves of patients with lung carcinoma, non-small cell lung carcinoma patients. And there are some survival curves, and I would like to show you this red line. It's a combined chemo immunotherapy, and you see the survival of the patients is not the best. And when you look to the blue line, you see antibodies against VEGF, a pure antibody therapy, and the survival of the patients is much better. So sometimes these therapies are in the outcome unexpected, and this is what we are interested in. We try better to understand what happens in patients under such immunotherapy. Our laboratory focuses on the cellular diagnostics in tumor patients. This cellular diagnostics is very fast, so we can detect the cells within a few hours after the blood sampling. We detect the cells that are the target of the therapy for diagnosis, monitoring, or to detect minimal residual disease. We try to monitor stem cell therapies. This is very common in stem cell transplanted patients. We detect the therapeutic efficacy, for instance, the reduction of tumor load. The exclusion of infections is very important. We can detect infection-specific T cells, for instance, for CMV infection. We can analyze by flow cytometry double-strand breaks. This can be very important in the monitoring of irradiation of patients. We can monitor the cellular immune system to prevent harm from the patient by avoiding side effects. And we can analyze, of course, the specific fields of the immune dysfunction. To do this, there are some laboratory tests available. The main limitation of all these cellular tests is that we need fresh cells, living cells, and the pre-analytic time is limited to few hours up to 1 day. To analyze these cells, we can use microscopy. This is not so common. Flow cytometry is a most important method, and we can combine flow cytometry with cell culture, for instance, to do stimulation tests, but we also can use ELISpot assays, proliferation assays, or cytokine release assays to analyze the function of the immune system. The main method we use, and this is the flow cytometry. Today, I am not able to show you too much about flow cytometry. It's simply to say you need a single cell suspension. The markers of the cells you want to investigate have to be marked with fluorescent dyes. And today we very often do a multicolor analysis. There is a wonderful, more or less, handbook of flow cytometry. It has been published in European Journal of Immunology from Andrei Kozarista. And here is the link. It's free, available in the internet. Some of the flow cytometers we use today are shown here on this next slide. There are some companies very common in most laboratories in routine work are flow cytometers from BD and from Beckman Coulter. It's a diagnostic tool, but it's also relevant for research, and we use it for the monitoring of the disease and for the detection of adverse events. There's one point I'm not so familiar with because it's not the main topic of our laboratory. This is the detection of leukemia. For instance, here is shown a typical picture of a TALL with a smear and a typical gating pattern of the lymphoblasts and some markers that commonly can be detected on the surface of these leukemia cells. This is today an indispensable tool for the analysis of leukemias, but this is not done in my laboratory. We are interested in the function of the immune system around this. And for instance, this is a very common effect we find in patients. This is the modification of the cell composition by an anti-B cell antibody. I hope it will work so that I not go to the next slide. Here are the B cell, is a B cell gate, and this is empty. This patient got treatment against B cells, for instance, rituximab, and rituximab completely depletes the B cells. In a comparison patient, these B cells are present. This is the normal way you should find it in a healthy patient. Another cell population we are interested in are CAR T cells. CAR T cells are produced by genetic modification of T cells with an additional chimeric antigen receptor. And this receptor is targeted to, for instance, in this example, to the B cell receptor CD20. And here we show the typical flow cytometry picture, what we find when we analyze these cells in the production of these cells, because in the blood of the patient, we will not find 20% of CAR T cells. In most patients, we find only a very low percentage of the cells, but it's powerful enough to treat these patients successfully. Besides this detection of the CAR T cells, we are interested in the composition of the immune cells. And what most of laboratory physicians and specialists know is this so-called immune status, this common screening tube. But this screening tube detecting NK cells, NKT cells, B, T, and B cells is not really powerful because there are many populations that are not detected. We get no information about the T cell maturation. We don't get information about the new formation of T cells, the so-called RTEs, the mesenchymic emigrants. We have no information about gamma delta T cells, no information about B cell maturation, about plasma cells, and so we can really miss an immune dysfunction. To overcome this point, we developed flow cytometry panels for CAR T cells, and these panels also include additional information about the activation of the normal immune cells and the CAR T cells, checkpoint information, PD-1, CTLA-4, for instance, exhaustion markers of the CAR T cells, giving information about the efficacy of the therapy, T cell senescence marker for RTEs and for H cells, cytotoxic proteins, and a lot other information about the function of the immune system. And this way, also with a combination of Th1, 2, and 17 cells, we can well describe what happens. And now we have a statistical disaster. We have a lot of information we have to present in an adequate way. And this is, for instance, a heat map of a multiparametric analysis of the cells in a non-small cell lung carcinoma patient. We need special algorithms to understand what's behind this. Another application of flow cytometry in these patients is the detection of infection. Here you can see the detection of CMV positive cells by a tetramer staining. Tetramer staining is very common to show CMV positive T cells, but it's limited by some disadvantages. You need MHC matched reagents. The majority of MHC molecules is not available, and it's very common in viral infections, but for other targets, it's not possible to apply this. We developed in my laboratory tests to use STAT5 phosphorylation. This has been done by Michael Badr. He has been a PhD student from Syria. This is the intracellular signal transduction following a stimulation of a T cell, and IL-2 and STAT5 are central points. And when we use STAT5 phosphorylation shown here, then we find a very good correlation in the graphs D and E with a normal common detection of cell proliferation. But the main advantage is we only need 1 day for a good result. And this is very important, so we can find out how many cells react to T cells. And for instance, we investigated it here in patients with an anti-B cell treatment. And we are concerned about the risk of being infected with COVID. And in these patients, we investigated the phosphorylation of STAT5, and we can see that 6 months after COVID infection, normal healthy donors have a very clear pattern, and there is a very low frequency of phosphorylated STAT5. 6 months after COVID infection in healthy people, there is still a lot of T cells with the capability to defend COVID, and the vaccination response in a patient under anti-B cell therapy is serologically 0, but there are a lot of T cells able reacting against COVID. So we know these patients have a certain degree of protection. Another topic is the detection of checkpoint molecules. You know that checkpoint molecules are a target for tumor therapies. Here I show you some cells and the typical checkpoint molecules we can find. What is not so well known is that besides the T cells, also dendritic cells, B cells, or PMNs in this picture can be used to analyze checkpoint molecules in immune stimulation. And here, for instance, is shown that PMN express a lot of VISTA. VISTA is one of the checkpoint molecules with a high relevancy for anti-tumor response. To do most of this work is bound to in-house developed test kits. Here I show you one guideline we developed in our research community for the establishment of flow cytometry tests in routine diagnostics. And I think this is a very important tool, and it should be translated into clinical practice. Take-home messages are the flow cytometry is important in the diagnosis of tumors. Cytometry helps to monitor the effects of biologicals. Immunophenotyping is relevant to prevent infections. Immunophenotyping indicates characteristic changes in the immune function, and the majority of the assays we can apply in clinical diagnostics needs validation. To bring these technologies also to Egypt, I'm very glad to be a partner in the EM-CERT network. This is a network of some Egyptian universities together with colleagues from Greece, France, and Germany, and we develop a human capacity building project to establish an immune modulation certificate. For all of you that have some scientific data ready to publish, there is a special issue in the moment of cancers. It's a journal with an impact factor of about 6+. There is a special issue, flow cytometry analysis of cancer, and the deadline for manuscript submissions is the 30th April of 2022. And I would like to thank you, and I will be happy to take your questions.
Thank you, Professor Sack, for the very fruitful and valuable session. We will have a Q&A session at the end of the webinar.
Okay.
So right now, we were gonna proceed to the second session.
Thank you.
Thank you so much, Doctor.
Pleasure. So I stop my presentation.
So in our second session today, we have Professor Sameh Saad Ali. His session title is gonna talk about mitochondria in cancer and why metabolism matters. Professor Sameh Saad Ali is a professor of biophysics and the head of Tumor Biology Research Program at Cancer Children's Hospital 57357. He obtained his master's and PhD degrees from Tohoku University, which is ranked number 70 in the world. He spent 3 years as a postdoctoral fellow in the Technical University at Graz and Washington University in St. Louis. He returned to Egypt from the University of California in San Diego in June 2012 to participate in the establishment of Egypt's national project. Professor Sameh is the founding director of the Center for Aging and Associated Diseases at Helmy Institute of Medical Sciences at Zuwail City. Also a member of the Egyptian National Committee of Biochemistry and Molecular Biology, the Technical Advisory Committee, the Egyptian Academy of Scientific Research and Technology, and the Scientific Committee in the Egyptian Genome National Project. Professor Sameh was awarded the State's Excellence Award in Basic Sciences in 2020. His work produced over 60 highly cited articles in many high-impact journals. He also acts as an executive editor, and regular reviewer for tens of internationally reputed journals, including Cancer Research, British Journal of Pharmacology, and Journal of Biological Chemistry. In our second session today, Professor Sameh explains the position of mitochondria in cancer and how oncogenic signaling and tumor cell metabolism are closely interrelated. In this presentation, Professor Sameh will recapitulate the hallmarks of cancer transformations with some emphasis on modulation of metabolic pathways to face bioenergetics demands, especially in proliferating cancer cells. He will then show examples of the data showing the potential of different metabolic interventional strategies against cancer cells. Welcome, Professor Sameh, to today's session. You can start now.
Thank you so much, Dr. Lina, for the nice introduction and for inviting me, Dr. Samira. I'm really happy to be among your guests. And I should actually salute the efforts done by your institute, which is really very important, including funding of scientific research and holding such kind of meetings, which indicates deep understanding of the importance of scientific research to the development of our country and the whole world. So let me just say that the topic that I'm going to cover today is uncoverable within the allowed time limit. So I'll try to be very quick, and also due to the nature of the audience, I'm obliged to give some sort of introduction to the concept of metabolism and mitochondrial function in oncology and in cancer research. This is quite recent and active and hot topic that's implicating mitochondria as a target for therapy, for cancer therapy. And let me just give you a quick introduction. Can I share my, do you see my presentation?
Not yet, doctor.
Let me try to share. Can you see it now?
Yes.
All right.
Perfect.
Okay, so we are in business now. So, so let me start by giving you an introduction why metabolism actually matters in our fight against cancer. I'm currently working in one of the largest hospital in the world in terms of pediatric oncology, and the contents of this seminar today is going to be just covering a little bit on origin of cancer and hallmark of cancer. Then followed by why cancer heterogeneity is constituting a very, very tough barrier between us and between finding cures for cancer. Then I'm going to try to sell the idea that metabolic pathways can be helpful in handling and treating many types of cancer. And we'll give you an idea why targeting metabolism might be a good weapon against cancer. And if the time allows, I would give some examples of our results. So I know that the audience may be aware quite well with the origins of cancers, but it helps to recapitulate the current theories. And of course, everyone knows that oncogenes and tumor suppressor genes and their roles in tumor development are now currently taking most of the theoretical background of the cancer origin. However, recently, a new theory has been suggested, which is talking about stem cells repair errors, and it's bringing a lot of debate and maybe some sort of life and vitality to the field. However, chronic irritation or continuous irritation combined with genetic predisposition leads to accumulated mutations, and accumulated mutations may produce some genotypic changes that would enhance and stimulate fast growth and hyperplasia. And this is usually associated with metabolic reprogramming, because coping with this fast growth requires bioenergetics. And this metabolic reprogramming, if combined with host with impaired metabolic flexibility, meaning that inability to return back to the original status, maybe due to chronic diseases, aging, obesity, lifestyle, and or environmental factors, this leads to metabolic catastrophe, and the metabolic catastrophe leads to tumorigenesis and metabolic switches. And upon progression, of course, tumorigenesis leads to tumor formation and then invasion and metastasis. And under certain circumstances, this extensively leads to systemic inflammation and organ failure, leading to cancer-associated death. So this is what is, simply speaking, what is known about the origin of cancer. In the following slide, which is going to be a little bit too busy, it's going to be about the cancer hallmarks, because this would pave the way why we are using metabolic strategies to target cancer. So the first thing is that, of course, everyone knows that one of the hallmarks of cancer is the sustained proliferation. And the next one is how tumors are evading growth suppressors and also inducing angiogenesis. And angiogenesis is quite important to determine the metabolic mode of a given tumor, leading to the possibility of also migration and metastasis and invasion, and also the replicative immortality, which is resulting from the telomere shortening impairment. And finally, we have also some sort of resistance for cell death. However, very recently, there are some sort of emerging hallmarks, which include avoiding immune destruction and also tumor-promoting inflammation, which we talked about in the previous slide. And then this can lead to genome instability and mutation. And finally, the deregulating cellular energetics. So most cancer types are associated with deregulation of cellular energetics and metabolic pathways. Okay, so why instead, or despite of the huge and massive investment in finding cures for cancer, we are still feeling that this is really a formidable task? I think the reason is that we are dealing with cancer as a sort of condition rather than dealing with it as a living organism. In my opinion, one should really look at cancer as a sort of living organism. And it actually develops capabilities. It can be very, very dynamic in facing drugs and developing drug resistance. And it is very also important to note the tumor heterogeneity, which can be a very, very bad barrier against us and finding a generic sort of treatment for cancer. So to give an example, this is an example published in the literature about how with time, with a short period of time, short span of time, we are seeing some sort of metastasis. And from these different sites, you can see that these different sites are providing very different heterogenic genetic fingerprint. Even on the level of cells, even individual cells, they are showing a profile of mutations and accumulated mutations that are really different. So it is quite tough to design a treatment. So now, if we are faced with tumor heterogeneity, and the complexity of this is probably resulting from a continuous talk between tumors and between the environment, there is no way that I can get a kind of biopsy and try to find ways to cure it, because it all depends, the nature of this kind of tumor will depend on the location where I get this kind of biopsy. So one has to think and be more creative about finding ways to deal with cancer. And on the other hand, heterogeneity leads to resistance. And resistance means that we are having a moving target. It's a dynamic target. So the biomarkers are very hard to identify and very hard to rely on, because this is a moving target. So the new therapeutic strategy should really start to because this is a living organism, you find ways that are very, very dynamic and also like a living organism as well. That's why we are focusing on the immune response itself and maybe immune therapy. But in the same time, if we should focus on something, we should focus on the stroma, try to resist the spreading of cancer, because dealing with the genomic instability is, in my opinion, is a lost fight. That's why we have to think about stroma, then we have to think about metabolism. And in fact, if you look at all of the hallmarks of cancer, you will find that there is a direct relationship, whether downstream or upstream, with metabolism. And that's why it makes a lot of sense to try to study metabolism and to try to find ways to handle the metabolic changes and the derangement that's happening in cancer. There are a lot of facts in the literature that actually supports this view, which include that oncogenic signaling and tumor cell metabolism are very, very related. Most of the mitochondrial genes are actually tumor suppressor genes, and it indicates that the involvement of mitochondria maybe is opposite to tumor health. So as a matter of fact, mitochondria is a cellular checkpoint that is critical for apoptosis regulation and probably also the response for therapy. Most of the therapies are trying to trigger apoptosis, which is mediated by mitochondria. So the conclusion is that probably cancer cell survival involves the minimization of mitochondria function. So the question is that, can we somehow try to manipulate metabolic pathways to induce some sort of tumor regression or not? And of course, to do this, you need to intervene with a very complicated pathways, metabolic pathways. This is an oversimplified picture that's indicating the major sort of metabolic pathways that are used in different cancer types, the glycolysis, and then mitochondrially mediated pathways, including glutaminolysis, lipid peroxidation, and also oxidative phosphorylation. And these pathways, essentially, you need to understand more about them before you can really go and try to intervene with cancers. So to do this, we have constructed in our hospital an advanced laboratory for metabolic studies, and this includes some cutting-edge instrumentations that would allow us to study mitochondrial respiration, reactive oxygen species, synthesis of ATP, mitochondrial membrane potential, calcium dynamics, and so on. And also, we have a system that would allow us for label-free cell counting, multi-mode reader applications, and drug discovery and immunofluorescence, and also live cell imaging. On the other hand, we have electron paramagnetic resonance spectrometer, which would allow us to study oxidative stress and membrane biophysics, protein structures, redox status, antioxidant capacity, and also free radicals. Finally, we have the Seahorse XF96, which is the top of the line system that can enable us to identify different metabolic pathways in a given cancer cells or cell to be attached. So the approach for us is to try, let me skip this one. The approach is a kind of reverse translational personalized approach where we take a biopsy or a sample from a patient, and then we do metabolic phenotyping. Of course, we should also take samples from different locations in the tumor, and then we do metabolic phenotyping. We try to establish primary cells, and then we can use these cells for metabolic targeting, developing drugs to target the prevalent metabolic pathway, and then developing more drugs and optimizing these drugs. And then you can try some preclinical models, including zebrafish and mouse models. And then you can try to pave the way for clinical trials for targeting metabolism to sensitize cancer cells for death, even with lowered doses of chemotherapy, or even individually to go together. And there are currently very few clinical trials along this direction. So let me just go in the few minutes that are remaining. Let me just give you an idea on what we do really. And in this case, we are targeting or comparing what happens in metabolism, both in triple negative breast cancer cells and hormone responsive breast cancers. As you know that the triple negative breast cancer the prognosis is much lower than the hormone responsive. And what we did is we actually used two cell models, one that represents triple negative breast cancers, which is MDA231, and another one that represents metastatic invasive ductal carcinoma, but hormone responsive. And we used these two cells we performed metabolic profiling and did the metabolic targeting. So by doing metabolic profiling, we actually measure the major pathways. The first pathway that we measure is the mitochondria-mediated metabolism, and we can essentially change the substrates to trigger and single out the different metabolic contributions, or we can measure the extracellular acidification rate that measures glycolysis. And from these results, we can see that the hormone responsive are more prone for or more inclined to use mitochondria, while the triple negative breast cancer cells are more using glycolysis, which gives us the idea, or gave us the idea to target glycolytic pathways. So we moved forward and tried to select some glycolytic inhibitor. This is iodoacetate, which is known to be an alkylating agent, and also it triggers GABTH, which is very important and very enriched enzyme in cancer, and it is important for glycolytic metabolism. We targeted these cells by metabolism, and in fact, after 4 hours, as expected, the cells that are using or relying on glycolysis showed a very strong cell death, whereas those that are not, they actually showed some resistance and resilience to the iodoacetate treatment. However, to our surprise, after 24 hours, this kind of behavior reversed, especially at lower concentrations, which told us that maybe there are something happening here. We went forward and we tried to study the treatment effects on the apoptotic signaling, which is using a PI and XMV by flow cytometry. And what we saw is that in case of the hormone responsive, at later times, they are more prone for cell death and apoptosis, whereas the triple negative, they still have survivor cells. At earlier time, the reverse is true. So we went forward and we also studied some genes that are responsible for mediating apoptotic pathways, and we found that the triple negative cancer cells, they do not express BAC and BAX and caspase-9 effectively in response to the therapy relative to the hormone responsive cells. We did also study the effect of treatment on the cell cycle by flow cytometry, and we found that although this treatment leads to growth arrest in case of the hormone responsive cells, especially in the G2 phase, this is not really happening in the triple negative breast cancer cells. So we went forward and tried to understand why this is happening. I'm not going to involve you in so much details. This paper is already published, and I will show you the title or the citation, so that if you would like to see some details, you can go for this. But we studied the instantaneous effect on inhibitions, and we found that the inhibition is very prominent in the beginning. After 4 hours, we can see that the hormone responsive cells, they were not affected early, but the triple negative breast cancer cells, they showed some sort of enhanced oxygen consumption rate. And then later on, they actually showed a very, very prominent oxygen consumption, which indicates that mitochondria probably is playing a very important role on the recovery of these cells later on. So we went forward and tried to study this phenomena, and we found that there are some sort of a resilient population that lasts in the triple negative breast cancer cells, and these are having some sort of mitochondria that still are active. So the idea is that probably mitochondria, this is a new population of the cells that actually have much more active mitochondria that help it to conserve these cells and to revive these cells. It is for triple negative breast cancer cells, there is a treatment that's now being under clinical trial, which is targeting PARP enzyme, which is a repair mechanism enzyme. And we wanted to see if really this treatment can lead to some sort of sensitivity to the PARP. The expression of PARP, we found that expression of PARP was essentially significantly higher in the triple negative breast cancer cells relative to the hormone responsive cells. And this probably explains the resilience and the ability of these cells to come back after this challenge. And we suggest here that probably it is important to combine PARP inhibition with also mitochondrial inhibition to increase the efficiency. There are a lot of other details. I'm not going to dwell into these, but essentially the take-home message here is that mitochondria might be an important target for combinatory therapy that would probably lead to some improvement in the current standards of care for facing certain types of cancer. We have plenty of other results regarding leukemia and also some sort of brain tumors, very rare brain tumors. And we think that metabolism and metabolic interventions may provide very interesting and intriguing sort of approach for this. And I'd like to finish my talk by providing I should really thank the group that essentially performed all the work. And I should also thank you for the invitation and thank you all for listening to my talk. Thank you.
Thank you so much, Professor Sameh, for the very fruitful session and for giving us an innovative aspect of cancer therapeutics.
Thank you very much.
So right now, we're gonna move on into the third session. Our first speaker, Professor Gamal Badr, is a professor of immunology and vice dean for graduate studies and research in the Faculty of Science-Assiut University. Professor Badr has a PhD in immunology with the best distinction, très honorable, from the Faculty of Medicine, Science-Assiut University, France. Professor Badr is also a coordinator of the International Relations Office at Assiut University. Professor Badr was awarded as one of the world's top 2% scientists by Stanford University, the best Arab researcher from the Association of Arab Universities, the State Encouragement Prize in Basic Science from the Academy of Scientific Research and Technology in Egypt, and awarded as a top reviewer from many high-esteemed Q1 journals. In our third session, Professor Badr discusses the synthesis of new thiazolidine compounds that mediated growth arrest of breast cancer cells. Professor Badr synthesized with his team 18 new compounds of thiazolidine 2,4-diones with his team and tested their anti-cancer effect against breast cancer cell lines. As well as non-cancerous breast epithelial cells, all the three compounds decreased the viability of breast cancer cells without affecting the viability of non-cancer breast epithelial cells. Using MTT assay and flow cytometry analysis, the compounds mediated apoptosis and growth arrest of breast cancer cells in a dose-dependent manner. Using human breast cancer cells isolated from patients, they found that the three compounds significantly mediated apoptosis of human breast cancer cells without affecting the survival of normal non-cancerous breast cells that were isolated from the same patients. Welcome, Professor Badr, and you can now start your session, please.
Thank you, Dr. Lina, for this presentation, and I would like to thank MARC for giving me this opportunity to present our recent work. So I have to share my screen now. It's okay?
Yes, doctor, you can go ahead, please.
Okay, so I will present my work entitled Synthesis of New Thiazolidine Compounds that Mediate Growth Arrest of Breast Cancer Cells. First of all, I think we are known that the most disadvantages of chemotherapy, which mediate destruction of many cells, including stem cells in the bone marrow, and hence it distracts the white blood cells, which is the immune cells, and also red blood cells. leading to anemia and infection by microbes, and also affect the hair follicles, mediating hair loss, affecting buccal cavity and mediating inflammation in the mucosa lining the gastrointestinal tract, and also destruct the cells that of reproductive systems, and also it mediates many damage for many cells from patient, and this different from patient to another patient. So thiazolidine compounds are very important compound or heterocyclic ring systems, which are widely used for many applications in medicine. These applications are used for treatment of pathophysiological conditions, including diabetes, diabetic complications, microbial infections, and also in cancers. The aim of our studies is to synthesize new derivatives of TZD compounds, and then monitoring their anti-cancer effects, and also to clarify the underlying molecular mechanism. So as Professor Sameh previously mentioned, that the two-cell line MCF7, which is hormone receptor, hormone dependent, and MDA-MB231 is non-hormone dependent. These two different breast cancer cell line we used to monitor the effect of these 80 new derivatives on these two cell lines, but also not only to this breast cancer cell line, we used MCF10, which is non-cancerous breast epithelial cell line, and just to monitor if this compound could induce apoptosis or growth arrest of cancer cells without affecting the viability of normal cells. And this is the most critical point, or this is the most side effects of chemotherapy. Then we isolated breast cells from patients who were subjected, 10 patients who were subjected to mastectomy, and also we isolate from the same patient breast epithelial cells that are non-cancerous but are normal cells. We use different techniques, including MTT assay, flow cytometric analysis, and Western blot analysis to monitor the underlying underlying molecular mechanism. So firstly, from the 18 compounds that we tested, we found that only 3 compounds, which known as compound 7, 9, and 5, which have anti-cancer effect on both MCF7 and MDA cell line. So these compounds mediate growth arrest of the two cancer cell line without affecting the viability of non-cancerous breast epithelial cell line, with an IC50 equal 1.3 micromolar. This was done using the MTT assay. Then we isolated, this is very important, we isolated breast tissues from 10 patients with breast mastectomy, and also from the same patient, we isolated normal, healthy breast epithelial cells. And here, the two cells are either untreated or treated with the 3 compound 7, 9, and 5. We found this, that in the normal breast epithelial cells, either the cell were untreated or treated by the 3 compound, there is no induction of apoptosis, as shown from one representative experiment by flow cytometry using double staining with annexine V and propadium iodide. However, for the breast cancer tissue cells, when the cell was untreated with this compound, the percentage of apoptosis was nearly 30%. But once these cells treated with the 3 different compounds, these cells from 70 to 80% undergoes apoptosis by double staining using annexine V and propadium iodide. This is one representative experiment, but accumulated data from 10 different experiments showed that normal priest cells are not affected by the treatment of these different compounds. However, the priest cancer cells, when treated with these three compounds, we found nearly from 70 to 90% of the cells undergoes apoptosis. And this indicates that these compounds are most specific inducer of apoptosis in the priest cancer cells without affecting the viability of normal priest cells that isolated from the same patient. Then we go to the molecular mechanism. As Professor Sameh mentioned that the signaling pathway PI3 kinase AKT and mTOR is very important and critical for the survival and the metastasis of cancer cells. We focus to investigate the expression and phosphorylation of many factors inside the cells. So we focused firstly on the signaling pathway PI3 kinase AKT mTOR. So we detected the phosphorylation of AKT mTOR and the expression levels of vascular endothelial growth factors and hypoxia-induced factors. If we see here clearly the normal, this is normal control tissues from 5 patients, we found that the phosphorylated AKT is not obviously cleared in the control non-priest epithelial cell. However, for the priest cancer cells, we found the cells when untreated by these 3 compounds, the cells have a very high phosphorylated rate of phospho-AKT as compared to control priest epithelial tissues or cells. This indicates that the phosphorylation of AKT is very important and critical for the survival, for the proliferating of cancer cells. And this is one representative experiment. Then we normalized the phosphorylation levels of phospho-AKT to the expression level of total AKT, and accumulated data from 10 different patients revealed that the normal priest cells are not affected in the term of phosphorylated AKT when they are untreated or treated with these 3 compounds. In contrast to these normal priest cells, the priest cancer cells that isolated from the same patient, the cell have a very high and apparent phosphorylation of AKT. But when treated by the 3 different compounds, 7, 5, and 9, the phosphorylation of AKT was significantly decreased by nearly 3-fold. This is a phosphorylation of AKT. Similar data was found for the phosphorylation of mTOR, and this means that the treatment of cancer cell with the 3 compounds mediate significant decrease in the phosphorylation of AKT and mTOR, and then this is planted the signaling pathway PI3 kinase AKT mTOR. For the signaling pathway vascular endothelial growth factor and HIF-1 alpha, which is very important for the metastasis, we found that the priest cancer cells have a very high expression levels of vascular endothelial growth factors and the HIF-1 alpha. But when these priest cancer cells treated with the 3 different compounds, 7, 9, and 5, the expression level of vascular endothelial growth factors and the HIF-1 alpha was obviously decreased. This is one representative experiment, and then we normalize the expression level of vascular endothelial growth factors and the HIF-1 alpha to the total loading control beta-actin, and the accumulated data from 10 different patients that subjected to mastectomy revealed that treatment of breast cancer cells with these 3 compounds significantly decreased or blunted the expression level of vascular endothelial growth factors and HIF-1 alpha. Additionally, the BCL2 family members are very important proteins that play an important role for the metastasis and survival of cancer cells. And now I think in the there is different trials to use antibodies to blocking the PCL family members for treating different type of cancers. So if we look for the expression of the 3 protein BCL2, BCL-XL, and MCL1, these are anti-apoptotic proteins. However, there are another proteins is known as pro-apoptotic proteins, including PAC-PAX and BIM. So if we found here for the first experiment, we found that the normal control breast epithelial cells have normal expression of BCL2, BCL-XL, and MCL1. But the breast cancer cells isolated from the same patient have a very high expression of these 3 proteins, which is anti-apoptotic protein. That means these cells have a high survival rate as compared to the normal cells. But once we treat these breast cancer cells with the 3 different compounds, 7, 9, and 5, we found that the expression of BCL2, BCL-XL, and MCL1 was obviously decreased. This one representative experiment, however, accumulated data from 10 different experiments revealed that the expression levels of BCL2, BCL-XL, and MCL1, which are anti-apoptotic proteins, are significantly decreased when we treat the breast cancer cells with the 3 different compounds of thiazolidine. This in contrast to what we found in the normal breast epithelial cells that were isolated from the same patient, which are not affected by for the expression of these anti-apoptotic proteins. In contrast, the pro-apoptotic protein, PAC-PAX, and BIM are significantly increased in the breast cancer cells after treatment of these breast cancer cells by these 3 different compounds. This one representative experiment revealed that treatment of breast cancer cells with these 3 compounds obviously increased the expression of PAC-PAX and BIM. And accumulated data from 10 different patients revealed that the expression levels of pro-apoptotic proteins were significantly upregulated in the breast cancer cells when treated by the 3 different compounds. So the conclusion of our work is that thiazolidine compounds 5, 7, and 9 mediate growth arrest of breast cancer cells without affecting the viability of normal breast cells through the following mechanisms. Firstly, induction of apoptosis in breast cancer cells without affecting the survival of normal cells. Second, reduction in the expression of the phosphorylation of PI3 kinase AKT-mTOR signaling pathway, and inhibition of the expression levels of vascular and cellular growth factors HIF1 alpha. Additionally, it alterates the expression levels of PCL family members and mediating growth arrest and apoptosis of cancer cells. However, unsolved issue in this work. So we used only breast cancer and we treat breast cancer cells with these compounds. So we should use another type of cancer cells like prostate cancers. Secondly, we used or we isolated breast cancers from patients who subjected to mastectomy, but we need to isolate breast cancers from different patients with different grade of breast cancer. Finally, we should use genetically modified animal models to investigate the in vivo effects of these compounds using animal models. And finally, we will use, after trying all these factors, we will try to apply this in human trials. So thank you for your attendance.
Thank you very much, Professor Gamal.
Thank you.
Thank you, Professor Badr, for sharing with us the very promising results of your research, and thanks to all the speakers of today's sessions. So for the next 5 minutes, we will let the participants now share with us their questions to the speakers in the question box.
Ina?
Yes, doctor?
Just in case when nobody of the participants can ask questions now, could you forward our contact so that we could reply the questions later on?
Sure, doctor. We will share your email. Is it okay that we share your email?
Yes, of course. Yeah.
Okay. Are the other speakers okay with this option too?
Yes.
No problem.
Okay, we will wait for a further few minutes more. So if anyone can have any questions.
I have one question for Professor Sack, if it's possible.
Of course.
Yes. I would like to ask about if you did detect the phosphorylation using phospho techniques, the phosphorylation of AKT, and also the expression of CD107 on the tetramer positive cell, T cell.
This phosphorylation assays depend on the presence of flow cytometry suitable antibodies. We established it for ERK, for STAT factors, and also for interleukin-2 transcription factors, but not for the factors you mentioned. But when you have a specific question, we could try it because we have a lot of experience with it. And I also hope to be later in this year in Egypt, so we could exchange our experience.
Okay, thank you.
So sometimes the antibodies for the Western blot work well in flow cytometry, and sometimes it doesn't work at all. So I cannot predict it.
The expression of CD107 on the tetramer positive cell, or CD127. 127.
127. It's a
IL-2 receptor.
127 is no. 25 is an IL-2 receptor. 127 is an IL-3 receptor.
IL-7 receptor.
IL-7 receptor.
Yes.
We never tried it, but I can do so. It's in interesting field. Do you want to target the regulatory T cells?
Yes, because this is a marker of long survive for the tetramer positive cells. So it can give a good idea about the tetramer positive cell.
Yeah, I think this is a good idea because these phosphorylation processes are very fast. And when you detect it in flow cytometry, you can predict what happens with the cells at a very early stage. And this is what was of interest for us, because we can do a proliferation assay, but it takes up to 1 week, you know, from mixed lymphocyte culture, it can take up to 7 days. But for us, this is very late, and we try to get the result in 1 day. So CD127, okay, yes. I will check it.
Thank you.
Pleasure. Which university are you from?
From Science-Assiut University.
Okay. Thank you.
Thank you.
Okay, thank you very much for your time and effort, and it was really great having you and having this wonderful presentation. Lina, would you like to say anything at the end?
No, we would like to thank you all professors for your valuable input in cancer research and your valuable work. We were very happy today to have you with us, and we hope we have future collaborations in the future, inshallah. So thank you so much, and this is it for our webinar for today.
Thank you very much for the invitation. Shukran.
Thank you so much for the invitation and for the activity. Thank you so much.
Goodbye.
Thank you.
Good evening.
Thank you
very
for
much.
the participants. Have a good night, all.
Good night.
Thank you.
Goodbye.
Goodbye.
Thank you so much. Goodbye.
Goodbye.